Today, we're going to start off with cell culture basics. Selecting the appropriate cell lines. Consider the following criteria for selecting the appropriate cell line for your experiments. Species. Non-human and non-primate cell lines usually have fewer biosafety restrictions, but ultimately, your experiments will dictate whether to use species-specific cultures or not. So in our lab, we use mouse and human cells. For this lab, we'll be using mouse. Functional characteristics. What is the purpose of your experiments? For example, liver and kidney-derived cell lines may be more suitable for toxicity testing. Finite and continuous. While choosing from finite cell lines may give you more options to express the correct functions, continuous cell lines are often easier to clone and maintain. So yeah, finite are the ones that you only get a few passages out of them, so it ends up being very expensive to use those. And continuous are the ones where you just buy one vial and you just, they just divide like indefinitely. So they become a lot cheaper because you can use the same vial for years. Normal or transformed. Transformed cell lines usually have an increased growth rate and higher plating efficiency. They're continuous and they require less serum and media, but they have undergone a permanent change in their phenotype through a genetic transformation. Growth conditions and characteristics. What are your requirements with respect to growth rate, saturation density, cloning efficiency, and the ability to grow in suspension? For example, to express a recombinant protein in high yields, you might want to choose a cell line with a fast growth rate and an ability to grow in suspension. Other criteria. If you're using a finite cell line, are there sufficient stocks available? Yeah, you need to buy like several vials. Is the cell line well characterized, or do you have to perform the validation yourself? So for validation, they're just saying like if this cell line is supposed to express CXCR4, then you need to stain for that protein and show that this cell line actually expresses that protein. So that's one type of validation experiment. If you're using an abnormal cell line, do you have an equivalent normal cell line that you can use as a control? Is the cell line stable? If not, how easy is it to clone it and generate sufficient frozen stocks for your experiments? Acquiring cell lines. So you can actually take out like cells from a mouse yourself, or you can just purchase them from a company. Purchasing them is expensive, but that's what we did. Adherent versus suspension culture. There are two basic systems for growing cells in culture. As monolayers, that means that it's just one layer of cells on an artificial substrate. That's an adherent culture, so the cells attached to the bottom, or free floating in the culture medium. That's a suspension culture. The majority of the cells derived from vertebrates, with the exception of hematopoietic cell lines and a few others, are anchorage dependent and have to be cultured on a suitable substrate that is specifically treated to allow cell adhesion and spreading. Tissue culture treated, however, many cell lines can also be adapted for suspension culture. Similarly, most of the commercially available insect cell lines grow well in a monolayer or suspension culture. Cells that are cultured in suspension can be maintained in culture flasks that are not tissue culture treated. But as the culture volume to surface area is increased, beyond which adequate gas exchange is hindered, usually 0.2 to 0.5 mils per centimeter squared. So this is important. The medium requires agitation. This agitation is usually achieved with a magnetic stirrer or rotating spinner flask. So basically, we have to change the media for some cells like every day, and for other cells, it's every 2 or 3 days. So sometimes people don't want to change the media every day, and so they'll add like 2 or 3 times the volume of media that you're supposed to. And unfortunately, your cells can die because there won't be any gas exchange, so they won't be receiving oxygen unless you have like a stir or a spinner type thing, which we don't. So yeah. Okay, and it looks like hematopoietic cell lines are the ones that they're in suspension culture, and the rest of the ones that we usually use are adherent cultures. So adherent culture, this is appropriate for most cell types, including primary cultures. It requires periodic passaging, but allows easy visual inspection under the inverted microscope. Cells are dissociated enzymatically. We'll be using trypsin. And basically what this does is it breaks apart the connections between the cells, and then it detaches the cells from the bottom of the dish so that you can resuspend them and then reseed them to expand or for experiments. And this way you can have like a homogeneous layer on your plate, so that they can start growing everywhere. And then growth is limited by the surface area, which may limit the yields. This requires tissue culture treated vessels. Yep. Okay, and then for suspension culture, it's appropriate for cells adapted to suspension. These are few cell lines, like the hematopoietic ones. They're easier to passage, but they require daily cell counts and viability determination to follow growth patterns. Okay, it does not require enzymatic or mechanical dissociation because they're already pretty homogeneous. Growth is limited by the concentration of the cells in the medium, which allows easy scale up. They can be maintained in culture vessels that are not tissue culture treated, but they require agitation. This is used for bulk production, batch harvesting. Okay, the culture medium is the most important component of the culture environment because it provides the necessary nutrients, growth factors, and hormones for cell growth, as well as regulating the pH and the osmotic pressure of the culture. Although initial cell culture experiments were performed using natural media obtained from tissue extracts and body fluids, the need for standardization and media quality, as well as an increased demand, led to the development of chemically defined media. The three basic classes of media are basal media, reduced serum media, and serum-free media, which differ in their requirement for supplementation with serum. So we needed to standardize the media types so that if I perform experiments here in California and then someone else performs the same experiments in New York, we're using the same exact things, so we should be able to get these same exact results. Serum is vitally important as a source of growth and adhesion factors, hormones, lipids, and minerals for the cell culture of cells in basal media. So serum is something that we normally add to a basal medium. So the basal medium is just like the basics, and then serum is added for additional stuff. And it has the adhesion factors, growth factors, hormones, lipids, minerals for the culture of cells in basal media. In addition, serum also regulates cell membrane permeability and serves as a carrier for lipids, enzymes, and micronutrients and trace elements in the cell. However, using serum in media has a number of disadvantages, including high cost, problems with standardization, specificity, and variability, and unwanted effects such as stimulation or inhibition of growth and or cell function on certain cell cultures. If the serum is not obtained from reputable sources, then contamination can also pose a serious threat to successful cell culture experiments. Serum is taken from animals, so it's batch dependent and it's batch variable. So if you take serum from a cow, then it's going to be a little bit different. The composition of it is going to be a little bit different than serum taken from a different cow across the country. And so those are some of the negatives of using serum. Basal media. The majority of cell lines grow within basal media, which contains amino acids, vitamins, inorganic salts, and a carbon source such as glucose. But these basal media formulations must be further supplemented with serum. Reduced serum media. Another strategy to reduce the undesired effects of serum in cell culture experiments is to use reduced serum media. Reduced serum media are basal media formulations enriched with nutrients and animal-derived factors, which reduce the amount of serum that is needed. Serum-free media circumvents issues with using animal sera by replacing the serum with appropriate nutritional and hormonal formulations. Serum-free media formulations exist for many primary cultures and cell lines, including recombinant protein producing lines like CHO, various hybridoma cell lines, insect lines, and for cell lines that act as hosts for viral production. Okay, so there's a lot of cell lines where people have already established what formulation they need, so then you don't need to use serum because they already determined you need this much of this product and this much of that product and this much of that product, and then it's perfect. But for other cell lines, we don't have that. No one has established it yet. So for those, you have to use serum. One of the major advantages of using serum-free media is the ability to make the medium selective for specific cell types by choosing the appropriate combination of growth factors. Okay, here's a table that covers the advantages and disadvantages of using serum-free media. So for disadvantages, the requirement of cell type specific media formulations. So like I said, some cell types, we already know the formulations that are required, so we can just make them and then it's perfect. But for other cell types, we don't know what the formulation is. So we need to establish it, or we just need to use serum. Another disadvantage is that we need higher degree of reagent purity. So that means that it's going to cost more money. Anytime you need to order things from a company, the higher the purity that you need, the more expensive that it's going to cost. Also, you get slower growth. So those are the 3 disadvantages of using serum-free media. Now, the advantages of using serum-free media are that you have increased definition, more consistent performance because you don't have that batch-to-batch variability. You have easy purification and downstream processing, precise evaluation of cellular functions, increased productivity, better control over physiological response, enhanced detection of cellular mediators. Okay. pH. Most mammalian cell lines grow at a pH of 7.4, and there's very little variability among different cell strains. However, some transformed cell lines have been shown to grow better at slightly more acidic environments, pH of 7 to 7.4, and some normal fibroblast cell lines prefer slightly more basic environments, so 7.4 to 7.7. Okay. CO2. The growth medium controls the pH of the culture and the buffers the cells in culture against changes in the pH. Usually this buffering is achieved by including an organic like HEPES or CO2 bicarbonate based buffer because the pH of the medium is dependent on the delicate balance of dissolved carbon dioxide and bicarbonate. Changes in the atmospheric CO2 can alter the pH of the medium. Therefore, it is necessary to use exogenous CO2 when using media buffered with a CO2 bicarbonate based buffer, especially if the cells are cultured in open dishes or transformed cell lines are cultured at high concentrations. While most researchers usually use 5 to 7% CO2, 4 to 10% CO2 is common for most cell culture experiments. So yeah, our incubator is normally set to 5% CO2, and that way we don't really have to worry about this. However, each medium has a recommended CO2 tension and bicarbonate concentration to achieve the correct pH and osmolarity. Yeah, we don't worry about that because our incubator takes care of that. Temperature. The optimal temperature for cell culture largely depends on the body temperature of the host from which the cells were isolated, and to a lesser degree on the anatomical variation in temperature. Overheating is a more serious problem than underheating for cell cultures. Therefore, often the temperature in the incubator is set slightly lower than the optimal temperature. Most human and mammalian cell lines are maintained at 36 to 37 degrees Celsius. Yes, our incubator is always set to 37 degrees Celsius. And then we have insect cells and avian cells. Cell morphology. Regularly examining the morphology of the cells in culture, like their shape and appearance, is essential for successful cell culture experiments. In addition to confirming the healthy status of your cells, inspecting the cells by eye and a microscope each time they are handled will allow you to detect any signs of contamination early on and to contain it before it spreads to other cultures around the laboratory. Signs of deterioration of cells include granularity around the nucleus, detachment of the cells from the substrate. And so as cells detach from the bottom, they actually become more circular and then they start to float. And that's how you know that it's not a good sign. Signs of deterioration may be caused by a variety of reasons, including contamination of the culture, senescence of the cell line, or the presence of toxic substances in the medium, or they may simply imply that the culture needs a medium change. Allowing the deterioration to progress too far will make it irreversible. So yeah, you just got to keep an eye on them. They got to be like your babies. Mammalian cells. Variations in mammalian cell morphology. Most mammalian cells in culture can be divided into three basic categories: fibroblastic, epithelial-like, and lymphoblast-like. Fibroblastic cells are bipolar or multipolar and they're elongated. They also are attached to a substrate at the bottom of the dish. Epithelial-like cells are polygonal in shape with more regular dimensions, and they also grow attached to the bottom of the dish in discrete patches. And then for the lymphoblast-like cells, those are spherical in shape, and they usually grow in suspension culture. So other cells also grow depending on what their function is. So we have neuronal cells, and they exist in different shapes and sizes, but they can roughly be divided into two basic morphological categories. We have type 1 and type 2. Type 1 has long axons, and they're used to move signals over long distances. And type 2 has no axons. A typical neuron projects cellular extensions with many branches from the cell body, which is referred to as a dendritic tree. Neuronal cells can be unipolar or pseudo-unipolar, with a dendrite and axon emerging from the same process. Bipolar with the axon and single dendrite on opposite ends of the soma, the central part of the cell containing the nucleus, or multipolar with more than two dendrites. So here are some pictures of healthy 293 T cells. This is at 10x magnification or lower magnification, so you can see more cells. And then here we have higher magnification. This is 20x magnification. So you're zooming into one location within over here. And each of these areas is called a field of view. So whenever you capture one image with whatever microscope, that's a field of view. And then here we have 293F cells, and these are grown in suspension. And you can see that when the cells are floating around, they're a lot more circular. And again, this is 10x and this is 20x. Okay, here we have a guidelines for maintaining cultured cells. What is subculture? Subculturing, also referred to as passaging, is the removal of the medium and transfer of cells from a previous culture into fresh growth medium, a procedure that enables the further propagation of the cell line or cell strain. The growth of cells in culture proceeds from the lag phase, following seeding to the log phase, where the cells proliferate exponentially. When the cells in adherent cultures occupy all the available substrate and have no room left for expansion, or when the cells in suspension cultures exceed the capacity of the medium to support further growth, cell proliferation is greatly reduced or ceases entirely. To keep the culture at an optimal density for continued cell growth and to stimulate further proliferation, the culture has to be divided and fresh medium supplied. So if we let too many cells fill the bottom of the dish, like I had told you, confluency, let's say this is our dish, and confluency has to do with the percentage of the floor of the dish that is open, that doesn't have cells on it. So your dish can be 60% confluent, 70% confluent, 80% confluent, and then you just kind of guesstimate to the best of your ability. And again, subculturing just means you passage from one plate to another, and this will allow your cells to continue growing and dividing. Yeah, because if, let's say, we let this entire dish fill up with cells to like 100%, that's actually not healthy for them, and they might actually stop growing. Okay, so here we have cell density on the y-axis, and then we have days from 0 to 10 days. So the log phase, right here we have log phase growth from day 1, and then we subculture when it's over here at like day 5. Okay, I'm going to read the legend. This is a semi-logarithmic plot, and it shows the cell density versus the time spent in culture. Cells in culture usually proliferate following a standard growth pattern. The first phase of growth after the culture is seeded is the lag phase. Okay, so that's the lag phase. We're going to need to know these for our quiz, which is a period of slow growth when the cells are adapting to the culture environment and preparing for fast growth. The lag phase is followed by the log phase. Okay, so that's the log phase. That's the logarithmic phase. This is a period where the cells proliferate exponentially, and they consume the nutrients in the growth medium. When all the growth medium is spent, i.e. one or more of the nutrients is depleted, or when the cells occupy all of the available substrate, the cells enter the stationary phase. So when they run out of a nutrient, they go into the stationary phase, so they stop dividing. Or when they fill up the bottom to like 100% confluency, then they go into the stationary phase. That's the plateau phase, where the proliferation is reduced or ceases entirely. So this right here is the lag phase, this is the log phase, and then this is the stationary, right there, stationary phase. Okay, when should you subculture for mammalian cells? Adherent cultures should be passaged when they are in the log phase, before they reach confluency. Normal cells stop growing when they reach confluency. This is called contact inhibition, and it takes them longer to recover when they're reseeded. So that's a word we need to know, contact inhibition, when cells stop growing because they reached confluency. Also, you passage cells when they are in the log phase. Transformed cells can continue proliferating even after they reach confluency, but they usually deteriorate after about 2 doublings. Similarly, cells in suspension should be passaged when they are in the log phase, before they reach confluency. When they reach confluency, cells in suspension clump together. and the medium appears turbid when the culture flask is swirled. So that can make you think that there's bacterial or yeast contamination because the media is turbid, but really it's just that you allowed the suspension cells to grow too much, maybe past the log phase into the stationary. How do you know when your cells have exhausted all of your medium? So for mammalian cells, a drop in the pH, that means that the media color is going to change because of the phenol red indicator that it has. So it's supposed to be like a bright pink, but if the pH goes down, then it's going to change to like an orange and then a yellow. So the growth medium usually indicates a buildup of lactic acid, which is a byproduct of cellular metabolism. Lactic acid can be toxic to the cells, and the decreased pH can be suboptimal for cell growth. The rate of change of pH is generally dependent on the cell concentration. Cultures at a high cell concentration exhaust medium faster than cells at lower concentrations. You should subculture your cells if you observe a rapid drop in pH with an increase in cell concentration. Subculture schedule. Passaging your cells according to a schedule ensures reproducible behavior and allows you to monitor their health status. Vary the seeding density of your cultures until you achieve consistent growth rates. So I'm going to be feeding for you because you're only going to be there on Mondays, so I'm going to be doing this for you. Media recommendations for common cell lines. Many continuous mammalian cell lines can be maintained in a relatively simple medium such as MEM supplemented with serum, and a culture growth in MEM can probably be just as easily grown in DMEM, so MEM or DMEM. However, when a specialized function is expressed, a more complex medium may be required. Information. Okay. Yeah, so you can, every company, when you purchase your cells, will sell you the media that is appropriate for your cell culture. If not, they'll at least tell you like what you need to feed them. So here's an example. We've got 293 fibroblasts. They come from the kidney, and you want to feed them MEM with 10% FBS. A549, those are lung carcinoma, so lung cancer, and you need to feed them F12K with 10% FBS, and so on. Okay, so when it's time to passage, you have to detach the cells from the bottom of the plate, and there's different ways of doing that. So you can shake off the cells. This is a gentle shaking or rocking of the culture vessel or vigorous pipetting. Loosely adherent cells or mitotic cells. There's also cell scraping. There's like a little spatula like thing that you use to scrape the bottom of the dish, and it may damage cells, but for some cells you have to use a scraper. And then there's also enzymatic dissociation, and this is what we're going to be using. We're going to be using trypsin. This is used on strongly adherent cells. It helps the cells detach from the bottom of the dish, so you can pick them up and passage them into a new dish with new media. You also have trypsin, dispase, and TrpLE. Okay, so here is a general protocol for passaging adherent cells. So you remove and discard the spent cell culture medium from the culture vessel. You wash the cells. Because cells are kind of like fish in water. You know how fish like pee, and then you have to change the water or else the fish could die because the water could become toxic. So cells also, they secrete their metabolic waste into their plate, along where their food is. And so that's where you get lactic acid from. And lactic acid, once you get enough of it, it will decrease the pH, and it'll also become toxic to the cells. So that's why we have to wash, and we use PBS. Then we remove and discard the wash solution. And then we add the dissociation. So you can use TrpLE, but we will be using trypsin. and then we incubate for a few minutes at 37 degrees Celsius in the incubator, and then the cells will be detached. You can go ahead and collect them, and then you can centrifuge them, and then you can remove the trypsin, and then you can resuspend them in pre-warmed clean media, and then you go ahead and seed. Notes on subculturing adherent cells. Okay, just a couple of important notes. Densities lower than 20% confluency inhibits growth. Cells they like to be next to each other. So at 20% confluency, they're too far apart and they're not healthy. They won't grow. CO2 exchange is not recommended for insect cells. Maintain, okay. We don't care about the insect stuff because we don't. This is a protocol for passaging suspension cultures, but we're not going to be using that, so we don't have to worry about it.